My name is Anton McFadden. I'm currently a bio major at the Community College of Philadelphia. And while doing that, I'm also currently interning here at the Singh Center for Nanotechnology. My experience so far has been really great. Get to work with highly skilled professionals, pick their brain on nanotechnology and other sciences that can be immersed in nanotechnology. A lot of instrumentation is pretty fun, and the exposure is really good too. I never thought I would be here, so it's pretty cool. I'm currently working on microfluidic devices and their use in CAR T-cell therapy. With CAR T-cell therapy, vital T-cells are needed for the therapy to actually be successful. If they're non-viable, then once complex with the CAR gene, those T-cells probably won't be therapeutically efficient. So if we can assess the effects of it using microfluidic devices, that could be pretty useful and vital for immunology. So this is the introduction to my presentation, the utilization of microfluidic devices in CAR T-cell therapy. Here we can see the breakdown and the understanding of what the CAR gene is, which is genetically engineered from fragments of antibodies, which, complex with T-cells, help T-cells recognize specific surfaces of antigens. And antigens are any molecules or substances that induce and elicit an immune response within the host immune system. And those antigens can be toxins, viruses, bacteria, and even cancer cells. And then you have T cells or T lymphocytes, which are derived from leukocytes, an alternative name for white blood cells. T cells and B cells also work in tandem with one another to help foster immunity against foreign invaders such as the antigens I just mentioned. So altogether we have CAR T cells. These cells first are filtered out from the patient's blood sample and then given a CAR gene, and then we can grow and culture these T cells, and then once chemotherapy has finished, we can infuse such cells within the patient, and those CAR T cells can begin their immune capabilities. But this is a very complicated and arduous task, because with leukapheresis, a process that is used to filter out these T cells, you're taking so much of the patient's blood sample, which can lead to effects such as nausea, low blood pressure, fatigue. So we try to lower the susceptibility to some of these effects by perhaps using microfluidics. If we were able to create a microfluidic device that would better filter and facilitate the extraction and purification of the T cells, we can step away from leukapheresis instruments, which require too much of the patient's blood sample, and then use microfluidics, which can be a little bit more economic perhaps, more reproducible, and creates less waste, and also deters susceptibility to the aforementioned side effects of low blood pressure, fatigue, and nausea. This internship, I think, is for anyone who's curious about a field that is widely used in the manufacturing of a lot of products. If you're into technology, if you're into how the way things work and how certain infrastructures are made, anything on a cellular, molecular level, all the way on up to the human level, I think nanotechnology definitely has some aspects that are integrated into that. It's a very interesting field. You get to cooperate and work with a lot of intelligent individuals. I think it would be very vital and beneficial for anybody who has an inquisitive mind who's very curious about science.